inviting me here today to talk with you all. In preparing for this, I went to the website at BU and saw past scholars and current scholars that are in the REC program, a really impressive group. So I feel very, very honored to be asked to speak with you today. I will be sharing with you some of the work we've been doing in Alzheimer's prevention trials, and in particular, the work we've been doing that allow us to draw sound inferences about whether a treatment is working or not in early stage disease. The focus of all of this work has really been around what's meaningful, what's a meaningful change that we're observing, and whether that measure at the end of the day is going to stack up to the expectations that patients have, and whether that change matters in terms of what is most important to patients. And so the title of my talk changed just a little bit to be more precise, and that is Alzheimer's Prevention Trials Measuring Meaningful Change and What Matters Most to Patients. So before starting, of course, we'll do my disclosures. As mentioned, I am at Duke University. I oversee a state-funded effort called the North Carolina Registry for Brain Health. I receive NIH funding both for my work in the ADRC as well as my work at DCRI, the Duke Clinical Research Institute. I'm on the Safety Monitoring Board for the U.S. Pointer Study, and I currently do receive some money from the pharmaceutical industry, as noted here, in consulting.
I also just wanted to point, I think we still see the first slide.
Oh, okay.
Yeah, no worries.
Oh, it's advancing here. So let's see if I unplug this. Now it's thinking. Are you seeing my second slide now?
It's still, oh, now I am. Yeah, we see it in the presentation speaker view. So if there were notes, we could see those. So it's, we have the main slide and then there's the next slide off to the right.
Okay, let's see if we can do this better. How's that?
Yeah, that's good. We can see the second. That's perfect. Yep.
You just
Awesome.
seeing the slide?
Yep, just the slide. Perfect. We're on the
Okay.
affiliation one.
Okay, great. Sorry about that,
No,
everybody.
no worries. Thank you.
Okay, so in terms of getting started here, I want to set the stage by returning to our patients and what our patients expect. And this is a hypothetical patient. I'll call her Jane. And this is the kind of situation I think we're seeing ourselves in nowadays. You know, Jane is in her early 60s. In this case, I've made her just 60. She's worried because her mother passed recently in the last year at the age of 89, suffering from nearly 20 years with Alzheimer's disease. Her symptoms starting probably around the same age that Jane is right now. And so Jane has been noticing some little changes in her own memory for day-to-day events, mostly in terms of recalling names. And she doesn't know, is this normal? Is this just what to expect with age, or is this early Alzheimer's disease? And hearing about all the new medical advances and diagnosis and different biomarkers, should she go ahead and get herself tested? And if she should get herself tested, what does that mean? I mean, should she start a treatment and would it be worth it? What could she hope for in that context? Do these drugs that we've been hearing about, do they really alter in any meaningful way her risk of developing dementia? And so those are some of the questions that patients are asking us as they consider what's in the cards for them and what's meaningful for them in terms of how they approach their care. So today, what I'd like to do is I'm going to focus us on this early part of the Alzheimer's disease trajectory of disease. We're going to be focusing primarily on very, very early treatment and potentially even prevention of early Alzheimer's disease. I have really three objectives that I'll be discussing with you here today. We'll start by quickly reviewing the FDA approval process for medicines in Alzheimer's disease. We'll then move to a discussion of the challenges that we're facing as we move to this earlier stage in the disease trajectory. And then we'll be considering how best to communicate meaningful treatment effects to regulators, to clinicians, and most importantly, to patients and their families who may be affected by the disease. So let's start first with the first objective of where we are in terms of FDA guidance around all of this. So the FDA, of course, um, helps us to decide whether or not treatments are effective or not. And almost in lockstep with Alzheimer's disease being identified as a biological disorder, almost since 1906, you can see throughout the literature all of these different unproven kind of miracle cure claims that began to pop up in the popular press. Um, and typically, these claims are exploiting vulnerable patients and their families who are desperate for a treatment. And this I just pulled together from the last year of different headlines that I saw either on the internet or in social media feeds. And you can see here are some of the claims that were being made. You know, one simple breakfast food erases Alzheimer's, local man reverses Alzheimer's in 7 days. Alone cures Alzheimer's, making clinical trials obsolete. And miracle supplement that rebuilds brain cells, stopping dementia cold. No prescriptions needed. So, what's a consumer to do when you're reading this? When faced with a horrible disease like Alzheimer's, patients want to know what are the treatments out there? What's the evidence that supports them? Are they going to work? Are they safe? And are they available to me? So the FDA in 1938 was established through the Federal Food, Drug, and Cosmetics Act of 1938, and their role is really to oversee, as we all know, the safety of food, drugs, medical devices, and cosmetics. In order for a drug to be approved, and this would of course include Alzheimer's disease, a drug has to be established as being effective as well as being safe. And let's just focus on the first part of that equation. So, in order to show evidence of effectiveness, the FDA tells us that the drug must show clinical benefit from adequate and well-controlled clinical investigations that demonstrate the drug does what the label is claiming it does. And so, how is this done? Typically, it involves two well-controlled phase 3 trials, or it could potentially be one trial that's exceptionally persuasive, multi-center trial. Importantly, in order to demonstrate a clinical meaningful benefit, the trials should not only just show a significant effect and a positive outcome, but they should show that this somehow positively affects how patients feel, how they function, or how long they survive. And I bolded this here because I want you to hold on to this. This is the core construct behind meaningfulness. It's not enough to have a significant effect on a measure, on a clinical outcome measure. It must also have impact on patient health. And this is what feel, function, and survive are capturing in that definition. So, in Alzheimer's disease, in the symptomatic disease stages, we're talking here MCI and later stages of dementia. Traditionally, dual endpoints of cognition and function have been required in order to meet that efficacy criteria that I just outlined. And why is that? Why do we have to have both cognition and function endpoints? The reason behind that is to ensure that improvement on a cognitive measure, like a memory measure, isn't just a statistical change, but it's also accompanied by something meaningful, a functional benefit. The other side to that, though, that sometimes doesn't get mentioned is it's also so that any observed functional benefit can be attributed to a change in cognition and not to some other change, like a change in lifestyle or a change in medical conditions that might have been contributing to that functional deficit. So it's really to show that there's a cognitive change and a functional benefit occurring and that those are reliable. And so, what are the endpoints that have been used in the symptomatic stages? I just do this as a point of review. So, in the area of cognition, the commonly accepted endpoints here are the ADAS-Cog, or the Alzheimer's Disease Assessment Scale Cognition. For the mild stages of disease, those tend to be the ADAS-Cog 13 and ADAS-Cog 14. When we move to function, there are various measures of instrumental activities of daily living. Common ones that are used in clinical trials are the ADCS, Instrumental Activities of Daily Living, and now the newer Amsterdam Instrumental Activities of Daily Living, which you see pictured here. For cognitive functional components, the popular measure that's been used now for many years is the Clinical Dementia Rating Scale. There's a global score that you get from that measure. That's used often for staging disease from having no problems all the way to severe dementia, a rating of 3. But you can also get a continuous measure out of the Clinical Dementia Rating Scale, and that's called the Clinical Dementia Rating Sum of Boxes. And there's another scale that's a relatively newer scale called the Integrated Alzheimer's Disease Rating Scale, excuse me, which also gives a continuous score. Excuse me, I'm going to take a sip of water. Okay, I'm fighting a cold or the end of cold. Other measures that could be used in those trials are global rating, excuse me, global rating scales of overall change. And this would include the clinician's global impression of change that you see pictured here. This measure involves careful interviews of both the patient and an informant to rate a change from baseline in a variety of dimensions, including cognition, function, behavior, and mood. So these are the common endpoints that are used in MCI trials and mild dementia trials. So now, well, fast forward, about 20 years since those trials started, and we now have Alzheimer's disease biomarkers that are reliable and validated that are enabling an interventional shift to earlier stages in the disease trajectory. And this is offering us opportunities now to delay symptom onset or potentially even prevent disease altogether. So there's lots of opportunities here, and I put up the classic Clifford Jack model of the unfolding of Alzheimer's disease pathophysiology over time. This is sort of the temporal progression of biological disease. And until we had these validated biomarkers, our studies of symptomatic disease were occurring essentially in the window that you see shaded here in green. This is when symptoms were becoming apparent. And you can see that the disease has already progressed fairly significantly at the point where we were intervening. Now, with the biomarkers unfolding, we can measure the beginning stages of the disease defined as abnormal A beta aggregation occurring very early in the disease. You see to the far left in these figures. Later, we see amyloid PET becoming positive, followed by soluble forms of tau measured in CSF, later on tau PET imaging, and then we see volumetric changes occurring on MRI imaging, culminating again to the far right with detectable cognitive and functional change on conventional instruments that have been used in clinical trials. So while we were confined to observing treatments during this window, noted here, we now find ourselves in a position of moving our trials way up here into the early window when disease symptoms may either be completely silent or very ambiguous at best. So as we move into this prevention context and into the preclinical stages of disease, we have this great opportunity to potentially essentially cure the disease or at least delay the onset of symptoms by quite a bit. But we also are presented with some challenges. So what are those challenges? Well, the first one, I think, is a fairly obvious one, and that is, well, what are the endpoints that are appropriate at this stage of disease? The traditional or gold standard endpoints that I summarized earlier, they were developed for the dementia stage of Alzheimer's disease, and they're likely going to be very insensitive to detecting change in the early asymptomatic stage of Alzheimer's. Another problem is that clinical improvement is not really expected. Rather, we're expecting a delay in the onset or a change in the overall disease trajectory. So, what are the outcomes that are validated for that purpose and recognized by the FDA for that purpose? What will be a pervasive, persuasive definition of meaningful change in this new context? And here you can see what we expect to happen in a disease modifying prevention context. You can see up where I have the circle A that there's very little change immediately from the treatment. And in fact, when treatment has begun, there's no change. And only over time do we begin to see the placebo, the dark line, diverging from the treated line, which is the dotted line. And you can see that the overall trajectory is ultimately what's being changed with these disease modifying prevention treatments. Nonetheless, prevention trials began to launch, and some of the big trials started in about 2013. These trials were starting, but we didn't really have a clear roadmap at that point in time. Um, we didn't know what type of trial design would be appropriate in the prevention space. We didn't have a good idea of the endpoints, as I've just summarized. We didn't know what would constitute a convincing change in this context of delaying or changing the disease trajectory. Length of study we knew would probably be much longer than doing a standard trial in symptomatic patients. But how long would that study have to last? And what would post-trial data collection have to look like to make sure we could make statements that we had, in fact, prevented, which is a very large claim, or just delayed the onset of symptoms? So we were kind of flying here without a roadmap. These were the early studies that I mentioned that launched in 2013. So we have the Diane study, the Alzheimer's Prevention Initiative, the ADCS-A4 study, and then our own Tomorrow study, which I'm going to be talking about here in just a moment. So these studies, as they began to enroll patients and then later begin to read out, the field was rapidly progressing and starting to validate different approaches that could inform this question of what are the endpoints we need to use in the preclinical stages that will detect a change and will be useful and meaningful in making inferences about whether treatments are working. And then later, about 5 years after these studies had launched, we have the regulatory guidance that came out from the FDA to help us in designing our trials. And this was later refined in 2024. The goal here was really the agency was providing information that would help form the thinking on the diagnostic criteria we use, as well as the approach to clinical staging of disease, with the goal of informing appropriate enrollment for clinical trials at different stages of disease and the selection of appropriate endpoints at those stages of disease. So basically, this guidance outlined 6 different stages to Alzheimer's disease, 3 that are in the pre-dementia space and 3 that are once dementia is clearly present. We're going to focus here on the pre-dementia and in particular on the first 2 stages of disease. So stage 1 or preclinical disease, as the FDA defined it, is when patients are either essentially, well, they're asymptomatic, they don't have any subjective complaint at that point, and no functional or cognitive impairment that can be detected. They're defined by having very low A beta PET levels, as you can see listed here. And the agency recommends that both clinical outcome assessments and biomarkers be included in those trials. However, to show that the endpoint is effective, or the drug is effective, I should say, a change on an Alzheimer's biomarker could be sufficient to show that this is efficacious. To show that it's meaningful, a pattern of effects across multiple biomarkers would increase the persuasiveness of that argument. Now, when we go to stage 2, which is when subtle cognitive changes are detectable, the patients may either have subjective complaints or it may just be that very subtle changes are detected on neuropsychological evaluation, but there's no functional impairment detected at this stage. And as you can see, it's defined biologically by having A beta aggregation that's at 40 centiloids on PET imaging and the appearance now of positivity in the tau biomarkers. At this stage, cognition change in cognition alone may be acceptable over short periods of observation or shorter trials. Um, to make it meaningful, those changes on cognition, a pattern across multiple tests would increase the persuasiveness as well as large effect sizes. And having change in the predicted direction also on the Alzheimer's disease biomarkers could also be very supportive of that claim that this change in cognition is meaningful in changing the trajectory of the disease. So what are the endpoints that are now being used to judge efficacy in this preclinical space and what's considered meaningful? So now we have more information that's helping to inform this roadmap. With the collection of data across these different studies, the accepted endpoints for prevention trials now tend to be these measures of cognition that you see listed here, either the PAC battery or the Alzheimer's Disease Prevention Initiative preclinical composite. For the endpoints of function and global ratings, we have these different IADL measures, as mentioned before, as well as global ratings of change, also mentioned before. It should be noted, though, that when we're looking at that stage 2 or preclinical early subjective cognitive change or preclinical disease, very little change is really going to be detected in these measures of function and global ratings, particularly over these shorter time frames of study. And it's really cognition that's the most sensitive and moving during that part of the disease process. So how do we demonstrate meaningful effects on these cognition measures? One approach is to look at the effect size across multiple measures, as the FDA has noted in its guidance, and the other is to have evidence of biomarker changing to indicate that the underlying biological trajectory is also shifting as cognition is shifting. Another approach that has been suggested is really in the study design itself and building in meaningful measures within the study design, such as a time to event approach to indicate a delay in progression. And this is exactly the approach we took in the Tomorrow study, which is why I mentioned it earlier. Our study was again to look at time to delay the onset of mild cognitive impairment due to Alzheimer's disease. So you may not be familiar with the Tomorrow study. It started in 2013 and it concluded in 2017. It was a single study, so it was designed not to be two phase 3 studies, but a single multi-center, double-blind, randomized, placebo-controlled study of a small molecule, pioglitazone, that would be taken daily, orally, to delay the onset of symptoms that would be diagnosed as mild cognitive impairment due to Alzheimer's disease. So the nearly 3,500 people that were enrolled in this study were completely normal at baseline when they were enrolled. The sample was enriched for people who were at Alzheimer's disease risk based on an algorithm that include two genes, ApoE and TOM40. Hence, what looks like a misspelling in the name for tomorrow is really an indication of that gene, the TOM40 gene. And age is the other variable in the risk algorithm. That risk algorithm is going to be important in a moment, so hold on to that. As mentioned, it was a single global registration trial conducted here in the US and across a number of other countries. It was aimed at disease modification with this chronic low dose pioglitazone. The molecule is believed to alter mitochondrial function and metabolism. It also is having downstream effects on A beta and other molecules affected in Alzheimer's disease. The study was to go for 5 years, so this was a long study. And the reason for that long duration is because it was a time to event design, with the event being moving from normal to a diagnosis of very early mild cognitive impairment. And 5 years was projected to be the necessary time to arrive at 202 events in our high-risk groups, which were necessary to make a well-informed statistical decision whether whether the compound was working. Now, I want to point out that we didn't use biomarkers as a criteria for getting into this study, and this is because those biomarkers that we now routinely use in prevention studies, amyloid beta, for example, were not yet validated and approved by the agency, so we had to go with only clinical criteria, and we were using the 2011 criteria for mild cognitive impairment as our endpoint in this study. Now, this study was unusual in that it had two primary objectives. One was to qualify that biomarker that I told you to hold on to, which was comprised of the TOM40 genotype, the individual's ApoE genotype, and their age. And to qualify that, we were looking at whether or not that biomarker predicted who would go on to develop MCI due to Alzheimer's disease. In our placebo arm. So we're contrasting high-risk individuals on placebo to low-risk individuals on placebo to test that question. Of course, this is a clinical trial, so we were also evaluating efficacy of a drug. Drug here was pioglitazone. And so we were again testing the objective of whether or not the drug worked to delay the onset of MCI due to Alzheimer's disease. And in this case, we're contrasting individuals who are in the high risk arm. So high risk placebo against high risk treated, just like any other clinical trial is contrasting the two arms. This is shown here in this little infographic. You can see we have a starting population over on the left from all the people from 5 countries, 3 continents. We had, by and large, 24,000 people who were screened in order to randomize that nearly 3,500 subjects who were in the trial. Most of the individuals who were included in the trial were in the high genetic risk arm, and that's because those will be the people who are receiving either placebo or treatment. And then we have a smaller group of individuals in the low risk, low genetic risk arm in order to test the biomarker. And each of these contrasts are shown here. So in our high risk group, this is a standard treatment trial looking at the contrast between placebo and treatment in people who are at high risk of developing the condition. In order to test the biomarker, we're contrasting our 2 placebo arms here to see whether or not being high risk predicts a faster onset compared to low risk. So what did we find?